Hi, my name is Rachel Hagan and I am a final year PhD student at Liverpool John Moores University. Today I am going to talk about a research plan for a study which takes a psychophysiological approach in order to investigate whether taste status is predictive of implicit liking of bitter, astringent and chemesthetic compounds. So food choice is generally influenced by flavour, and the extent to which an individual likes a flavour is an important driver of both short and long-term food consumption. Individual differences in food preferences is partly determined by genetic taste status and inherited relative sensitivity to taste stimulation. This can be measured by the density of fungiform papillae on the tongue and also by measuring individual sensitivity to a bitter compound known as PROP. Genetic taste status is broken down into three categories: non-tasters, medium tasters and super tasters. Super tasters are known to have more fungiform papillae on the tongue than medium and non-tasters, and researchers found that not only do super tasters have an enhanced sensitivity to the bitter compound PROP, but this is also enhanced in sensitivity to chemesthetic stimulants such as capsaicin chili or cooling menthol, and also astringent compounds such as those found in tea and red wine. But results in these areas are mixed and more work is needed to fully understand this relationship. So to date, most research on hedonic responses to food has relied on subjective ratings. However, these can be influenced by demand characteristics and social desirability effects. The current research will therefore look to tackle these limitations by using psychophysiological measures to investigate implicit liking of various oral stimuli. We hypothesize that super tasters will display increased negative affect-related facial muscle activity, greater cardiac deceleration, and lower effective ratings to all stimuli than non-tasters. The study will be conducted in two phases. In phase 1, 120 female participants will be screened for PROP taster status. Non-tasters and super tasters will then be invited to phase 2 of our study. During phase 2, participants will be presented with bitter, astringent and chemesthetic compounds at both threshold and suprathreshold levels. Effective responses to these oral stimuli will be recorded using two implicit measures: facial electromyography and electrocardiogram. Subjective ratings will also be recorded for intensity and liking. There will be two blocks within this phase: a tasting block during which the psychophysiological measures will be recorded, and a tasting and rating block during which psychophysiological and also subjective measures will be recorded. We hope that the proposed study will help us to understand whether PROP taster status is predictive of implicit liking of threshold and suprathreshold concentrations of bitter, astringent and chemesthetic compounds. Thank you.